Welcome to today's BioPhorum webinar, an intercompany perspective on compatibility and in-use stability studies to enable the administration of biopharmaceutical drug products. I'd like to start by thanking Nettie for organising the webinar for us. It's fantastic to see so many of you on the call today, and although it would be lovely to hear from you all, you have been muted to maintain good audio quality for the time of the presentation. If you'd like to ask a question, we'd really encourage you to do so by typing it into the chat box during the presentation, and if you could indicate who the question should be addressed to, that would be fantastic. The panel of presenters will try and answer all the questions after the presentation in a Q&A session. This call is being recorded and will be made available along with the presentation deck sometime after the event on the BioPhorum website. I'd now like to invite all the speakers to turn their webcams on. Okay, so I'd like to welcome Doug Kamen from Regeneron and Kin Yan, also from Regeneron. We have George Krotz from GSK R&D and Brittany Mills from AbbVie. We also have Francisca Bormita from GSK SED and Nachu Narasimhan from Merck & Co Incorporated. And my name's Elaine Stokes and I'm a lead facilitator with BioPhorum. Can we go to the next slide, please, Nettie? We're going to start with an introduction to BioPhorum and explain how this work came about. Each presenter will then cover the different aspects of compatibility and in-use stability studies as shown on this slide. We'll round up with some conclusions before we lead into a Q&A session. Next slide, please, Nettie. BioPhorum creates an environment where the global biopharmaceutical industry can collaborate to accelerate their rate of progress for the benefit of all. The development group focuses on better and faster process development of biopharmaceutical medicines. We successfully connect companies and provide a space where subject matter experts can work together on best practice and sharing approaches. There are 14 work streams in the development group, one of which is the formulation and drug product development work stream. And I have the very great pleasure of facilitating this work stream and the discussions that ultimately led to this presentation. On the next slide, we can see that contributions do come from a whole range of different companies. And this is a composite view of opinions from the work stream and shouldn't be attributed to the individual positions of the participating companies. Since contributions are received from companies of various sizes, of different product portfolios and a big range of business models, we think that the survey results are a representative summary of overall industry practice. I'd now like to, on the next slide, and hand over to Doug Kamen from Regeneron, who will cover the introduction to this work. And I'll just pop my webcam off now.
All right, thank you very much, Elaine. Can we go to the next slide, please? Okay, so what are compatibility and in-use stability studies and why do we do them? Well, the first box gives the answer to that. Compatibility and in-use stability studies are typically conducted during the development of therapeutic proteins to ensure patient safety and product efficacy. They are performed to assess the stability and compatibility of biologic drugs with diluents such as saline or dextrose solutions, components such as IV bags, infusion sets, filters, needles, and syringes, and under conditions such as room temperature or refrigerated storage and light exposure, and physical stresses such as mixing, transportation, and pumping. The BioPhorum Formulation workstream wanted to understand industry approaches to these important studies. So we conducted 5 benchmarking surveys with responses from up to 28 member companies conducted between 2018 and 2019. The intention was to obtain a broad spectrum of responses. So what was the outcome? In addition to a lot of details on how different companies performed these studies, One of the major conclusions was the realization that there is no one set of best practices that covers every company's unique needs for specific clinical programs. Instead, we uncovered a set of practices, all of which have been successfully employed in clinical programs. Other companies can use these approaches or even develop new ones based on these approaches to suit their specific clinical needs. Okay, next slide, please. Okay, this is just a snapshot of our manuscript highlighting the collaborative nature of the work. And I just wanted to point out that this paper was written by 16 authors from 12 different companies. Next slide, please. Okay, so this slide breaks down the paper into the survey categories. A total of 87 questions were asked, including 9 questions on regulatory strategy and feedback, covering items such as compounding, microbial challenge studies, data submitted, and dosing methodologies. We had 20 questions on clinical in-use formulation, patient and site considerations such as formulation factors and administration methodologies. There was 14 questions on blinding, masking, and placebo approaches such as placebo strategies and matching to drugs, effectiveness of blinding, and masking administration. 25 questions were asked on study setup, execution, and reporting, such as designing studies, qualifying materials, and selecting conditions. And finally, 19 questions on clinical in-use stability and compatibility testing, mostly discussing details around analytics, such as methods, qualification, and analytical acceptance criteria. Next slide, please. Okay, and I just wanted to have a few definitions, just so we were all on the same page. We've defined compatibility and stability as we use these terms in the manuscript. And so for our purposes today, compatibility is interaction between the drug and materials used in the preparation and administration procedure. And in-use stability is how the drug behaves during the preparation and administration procedure and the response to stresses encountered. Okay, next slide, please. All right, so I'd like to now hand off to my colleague, Qingyan Hu, who is going to talk about regulatory strategy and feedback.
Thank you, Doug. So I'll be presenting the several questions that on topics that has not been addressed by the current regulatory guidance. Next slide, please. So the first question in the box was on in-use time of compounded product. The question is, have you received the regular feedback limiting the duration that a compound product can be held at ambient condition based on microbial risk? So on the graph, 6 out of responded yes for IV product and 3 out of 12 responded yes for sub-Q product. So this result at least confirmed that in-use time is of regular concern due to microbial risk. On the right side is about microbial challenging studies. When does your company typically conduct microbial challenge studies of dosing preparations? As you can see on the graph, most of the companies conduct the microbial challenge studies at late phase and a BLA stage. And on the bottom of the slide is the table on the level of in-use stability typically included in your regulatory submission. The responses were separated into development stages, early, late, and market authorization. And most of the company provides comprehensive details and data in the filing. But some companies, especially at early stage, only provide high-level summary for their in-use stability study. So the filing content really varies among the survey companies. Next slide. So here in the first box, surveyed material tested in the compatibility studies. When does your company test a broader range of MOC, the material construction, for clinical administration? From the graph, most of the companies started testing broader range of material at the later phase, BLA, or as required. There's only one response who tested broader range of material at early phase. In addition, most of the respondents do not avoid testing DHP containing material, meaning that it is part of the compatibility testing panel, and that even DHP is included, most of respondents do not test plus hazard leaching during the compatibility studies. So on the right is the number of drug product batches tested for the BLA submission, and 3 responded only 1 batch, and 7 considered majority responded 1 to 3 batches, and only 1 responded using more than 3 batches. So in addition to these responses, we also learned that 5 out of the respondents do not test the drug product with different ages. So on this slide at bottom is a question regarding dose preparation. Have you designed a dosing preparation methodology that regulators consider manufacturing as opposed to compounding, and how this was managed? So here, dose preparation really refers to complex dose preparation, such as intermediate dilution or using a new container. And 3 out of the responded yes, and those preparation is considered as manufacturing. They manage such dose preparation by using on-site QP release or using a pharmacy with a special license. The very last question in this section is on lab setting. So does your company conduct a compatibility study and test samples used to a GMP or non-GMP standard? Within responses, most companies use a non-GMP lab, so 11 out of 12. There are also 5 responded with GMP lab, so the practice really varies. Next slide, please. So here's a quick summary. Currently, the regular guidance on compatibility and usability is quite general and brief. Specific guidance on study design and expectations are very limited. So through the survey, we understand that compatibility practices and filing strategy vary among participating companies, specifically on the level of details for compatibility studies submitted to regular agency, drug product batches and ages for testing, GMPs versus non-GMP lab setting. But all seem to worked out. So yeah, so with this, I'm going to pass on to the next section, which is going to be presented by George from GSK. He'll be discussing the survey results on clinical in-use formulation, patient and site considerations.
Thank you, Qingyan. Next slide, please. So the first slide covers formulation considerations, which we asked a series of questions shown in the top left around the minimum or maximum pH, osmolality, or viscosity administered via the IV subcutaneous or even the IM route of administration. So starting with pH, based on the survey respondents, we have the lowest recorded value was pH 4 and the highest was pH 7.5. Looking at osmolality, we asked what was the maximum value that you've used. And interestingly, the lowest maximum value that was reported was 235 milliosmolar for the IV route of administration. And that wouldn't be surprising for that route, considering the products are often diluted in a isotonic commercial diluent. The highest values that were reported were 1100 or 1200 milliosmolar for both IV or subcutaneous routes of administration. So you can see that quite high osmolalities are oftentimes used. Looking at viscosity, we probed the highest and or average values that were assessed. For subcutaneous route of administration, that's where we would expect to see the highest viscosity, considering the constraints around the injection volume. The average range from 1 to as high as 20 that were used as an average value, and just focusing on the maximum value here, we saw 80 centipoise as used by at least 1 respondent. Now going to the bottom left, we asked whether there was any technology to increase your maximum subcutaneous volume. 4 or 7 mentioned that they used excipient technology, and hyaluronidase is most prominent there. 4 or 7 used device, such as a wearable patch pump, and 2 or 7 used subcutaneous infusion to increase the subcutaneous volume. What technology facilitated administration of your maximum viscosity? Again, could be a common constraint by companies. That's most commonly, or the most common response was to use a larger gauge or thin wall needles. And lastly, top right, we asked whether there were any specific buffers or excipients that were found to have tolerability issues. 8 out of 10 noted no tolerability issues amongst any composition that they evaluated. One company mentioned that some stinging was noted upon subcutaneous injection, and that was based on using a citric acid-based formulation. Next slide, please. We asked then a series of questions around patient considerations. First was, what was the typical upper volume limit administered as a single bolus injection via the subcutaneous route? Some companies mentioned as low as 1, and others exceeded 2 ml. Most commonly, value reported was 1.2 to 2 milliliters. And what was the maximum needle gauge used to administer a liquid product via the subcutaneous route? The needles were as large as 18 gauge and as small as 27 gauge. And the most common response was to use a 27 gauge needle to administer a subcutaneous product. And on the right, we asked a series of questions that were related to the number of subcutaneous injections, whether it was for single or repeat dose studies, and as well as the clinical phase, whether it was early, late, or commercial. And not surprisingly, we saw that the largest number of injections were administered during early phase clinical trials. So some reported as high as 5 or 6, and even for repeat dose studies as high as 4. Moving to later commercial, there we saw that in general, there were a reduced number of maximum injections. Most common responses were around 1 or 2 injections, although some companies reported up to 4 injections for a single dose. Next slide, please. And then we asked a number of questions around site considerations. We asked what was the maximum and or minimum infusion rate that your company has safely used for IV administration. And looking at the figure on the left, the maximum flow ranged from 1 to up to 10 ml per minute. The central value 4.2 corresponds to 250 milliliters administered over 1 hour. In terms of the minimum infusion rate, it ranged 100-fold from as low as 0.02 ml per minute up to 2 ml per minute. Again, the central value there, 0.2, corresponds to 10 milliliters per hour. We asked, what do you use a syringe pump for infusion for IV or subcutaneous administration? 11 of 12 routinely used for IV, 5 of 12 routinely used for subcutaneous administration. And lastly, we asked about the number of vials that you had asked a clinical site to pull for a single administration of your product. The range was 4 and as high as 30, with a median of 20 vials. And those vials were pulled oftentimes 6 out of 10 times in a single sterile empty vial. Next slide, please. And now I'll hand over to Doug from Regeneron, who will be covering blinding, masking, and placebo approaches.
Thanks a lot, George. Next slide, please. All right, so blinding. Blinding refers to a clinical approach to maintain the integrity of a study by keeping the identity of the active and placebo control unknown. 8 out of 10 companies have a blinding or masking strategy for drug products used in clinical trials for IV or sub-Q administration. And although 9 of 9 companies that responded allow unblinded pharmacists, only 1 of 9 allowed an unblinded administration staff. The companies seem to be much more open to the idea of unblinded pharmacists compared to the staff who are actually administering the drug. The plot below shows the responses for what would cause your company to use an unblinded pharmacist. And most responses were for differences in things like viscosity, reconstitution time, and color differences between the active and placebo. And when it comes to performing assessments to determine if active can be distinguished from placebo, 4 of 10 companies said that they always do these assessments, and 6 of 10 companies said that they sometimes do these. Next slide, please. Okay, so masking. Masking refers to the procedure used to prevent unblinding. 6 of 10 respondents use masking approaches to prevent patients from directly observing injections. 2 of 10 respondents respondents reported using a masking system for at-home self-administration as well. When asked if they ever used a tinted vial or syringe cover for blinding, 2 said that they always use it, 3 said sometimes, 3 said never, and 2 said that they would use it based on experimental data and risk. One company further elaborated that tinted vials are not preferred due to the challenge in evaluating visual appearance during release testing. When asked, does your company mask the intravenous infusion bags and lines with covers or similar, 3 responded that they always do, 2 said sometimes, 1 said never, and 4 said that this was based on experimental data and risk. Lastly, we have a table summarizing masking approaches indicated by respondents. In general, companies use some kind of cover, either colored or transparent or opaque, to mask the vial, syringe, or infusion. Next slide, please. Okay, so placebo. Placebo is a substance that has no active ingredient or therapeutic effect, but is used as a control in testing new drugs. Companies are about evenly split on whether they use a custom placebo containing the formulation buffer for both IV and sub-Q. For companies that use a platform placebo across programs, the most common solution is normal saline, with some using custom placebos to match the active. Most companies do not use viscosity modifying agents to match the placebo viscosity to the active, and only 2 companies indicated yes, and that was for sub-Q. And finally, just some final points. Other than a couple of exceptions where viscosity modifying agents were used, companies are really not inclined to add ingredients to custom placebos that are not found in the active formulation. And so some specific responses indicated that coloring agents or inert proteins, such as human serum albumin, are not used to match physical attributes of active formulations. Next slide, please. Okay, so now I'd like to hand it off to Brittany. Brittany's going to talk about study setup, execution, and reporting.
Thanks, Doug. Next slide, please. So I'm going to start with the considerations surrounding the study design. In general, clinical and use studies are completed 3 to 6 months prior to the regulatory filing with representative material. In addition, manufacturing process changes do not warrant additional studies, but 8 of 9 respondents stated that this is evaluated on a case-by-case basis. When it comes to the study design, 9 of 9 respondents stated that they have a predefined company strategy for the selection of the diluent and the material of construction. Diving a little bit more deeply into the material of construction, 10 of 10 companies responded that they qualify the contact material of construction and not specific suppliers. These materials can be tested independently or in combination. In addition, 7 of 10 companies stated that they will test clinical site-specific sets if they have not been priorly assessed in lab. When determining the study design, 9 of 9 respondents stated that the clinical team is consulted for input on the dosing route, as well as the methodology for the clinical and use study. The last detail on this slide is regarding pump preference. 6 of 10 companies responded that there was no preference when it comes to use of either a syringe or an infusion pump, whereas 4 companies said that syringe pumps are preferred. In addition, infusion pumps are preferred for later phase studies since they are more common and preferred commercially. Next slide, please. Next, we discuss infusion considerations. First off, in that first panel, you can see that there is a diverse set of options that were reported regarding infusion rate. 7 of 10 companies responded that they use a static hold and relevant rates. In addition, companies responded that they bracket minimum and maximum rates, only test the maximum rate, or only test the minimum rate. To determine the minimum infusion time, 3 companies responded that it is based upon the lowest dose, and an additional 3 companies responded that it is based only upon the time needed to collect the sample for analytical testing. When trying to mimic IV administration, the use of inline filters was surveyed. 5 companies stated that they routinely test inline filters, whereas the other 5 stated that it is based solely upon a risk assessment. When filters are tested, PES is the most common material used, whereas the 0.2 micron size is the standard filter assessed. In addition, the filter material and size evaluated will expand as the program progresses into the later stages of development. Next slide, please. This slide details considerations surrounding dose administration and storage. The first panel goes into the pooling container that was used. Both syringes or IV bags were mentioned as containers that can be used for pooling of multiple vials. In addition, one company responded that it may change depending upon the number of pooled units. For the material of construction, either glass or plastic were noted as to be used for the pooling container. When it comes to the overfill strategy, 6 companies responded that an equivalent volume of diluent is removed from the IV bag prior to drug product addition. 3 companies responded that the volume is only removed when the drug product volume is large. In general, though, the overfill is not removed since the full IV bag is administered, and there is also pushback from clinical sites to simplify the process and avoid any extra steps. The last consideration discussed on this slide is surrounding temperature conditions as well as additional stressors. As you can see in the table, room temperature conditions are the most often tested temperatures, whether this be under uncontrolled ambient temperature and light, or within strictly controlled temperature chambers, such as at 25 or 30 degrees C. When it comes to testing light, it is not the effective light exposure on prepared infusion solutions at 2 to 8 C is not evaluated. 5 of 9 companies even responded that protecting from light is not required, even in the absence of stability data. With regards to shaking, 3 of 10 companies responded that they simulate shaking to simulate transportation or to respond to clinical site questions. Next slide, please. Finally, respondents were surveyed for common practices when it comes to hold time recommendations. Hold time is regarded as the storage time after the infusion bag is prepared, or a combination of the storage time and administration time. In general, the stability studies are conducted for longer than is required for clinical dosing, with 24 hours being the most common. While the limits are primarily established based upon the chemical and physical stability data, ultimately the hold time is restricted due to the requirements from health authority agencies for the risk of potential microbial growth. As you can see in the table, approximately 4 hours is the most commonly reported hold time under ambient temperature and light conditions. Under refrigerated conditions, 20 to 24 hours for hold time was the most reported number. Next slide, please. So finally, how do we use all of this data? So the clinical and use data is compiled and guides the authoring of the instructions for the preparation and the administration of the drug product. These instructions are then provided to the Clinical Trial Operations Group, which is the group responsible for training of the clinical sites. Companies noted that training videos could be used, as well as on-site training, or a combination of both on-site and teleconference on a case-by-case basis. Next slide, please. Next, I would like to pass it off to Francisca from GSK, who will be presenting on clinical use stability and compatibility testing. Francisca?
Thanks, Brittany. Next slide, please. For the in-use testing piece, we have divided our discussion into 2 parts. The first part, I am going to focus on analytical method specific questions. We asked on the left-hand side, companies the status of the analytical methods they typically use. From the responses in the graph, you can see that during early phase, most companies utilizes either unqualified methods or in some cases qualified methods. And as program progresses towards the late phase, some companies then introduce validated methods. Companies did define when methods are not qualified, how they demonstrate fit for use. And they demonstrate fit for use by evaluating matrix effect, linearity, precision, and detectable range. The next question that we asked was on potency assay. So we asked, when and what type of potency assay does your company run, whether it's early phase or late phase? From the graph, the respondents indicated that during early phase, binding assays are preferred. And in the late phase, both binding assays and cell-based assays are typically performed. We did get responses 4 and 2. Companies did indicate that regardless of the phase of the program, they do not perform potency assay. On the right-hand side, we asked companies the selection criteria for choosing an assay to perform. The results indicated that there are factors such as concentration of the admixture or sample they have, capability of an assay, as well as companies utilizing their prior knowledge or risk-based approach in selecting the assay that they want to perform on the samples. From the table, we have listed generally performed assays on in-use samples, and the results indicated that regardless of the concentration of the sample, most of these assays are actually performed. However, we did notice that from the responses, assays such as reverse phase HPLC, hydrophobic interaction chromatography, and CESDS, these assays are not performed when concentrations are below 1 milligram per ml. We also got additional commentary regarding particulate analysis, where most companies indicated that for visual appearance, they utilize a light box to perform this test. And for subvisible particle, all respondents indicated that light obscuration technology is preferred, whilst flow microscopy technology are typically used for characterization of particles. Next slide, please. I will now hand it over to Nacho from Merck, who will be going over the second part of the in-use testing, where he will cover assay acceptance criteria, sampling plan, and test schedule. Nacho?
Thank you, Francisca. So this part of the survey, as Francisca said, has to do with the assay acceptance criteria and the test schedule and sampling plans. When the companies were asked about what acceptance criteria they use, 7 of the 13 respondents said they use concentration and use product specification as the criterion for the concentration. Although 5 of them, while they use concentration as a criterion, they use product specific criteria based on the concentration of the product they are testing. And you see on the right side, the ranges of the acceptance criterion that companies use. 7 of 13 said their criterion between 90 and 110%, while 3 of 13 used 80 to 120. The next acceptance criterion companies use are purity, while only 5 of 13 responded that they use observable or no observable change from the initial value of the purity assay. 12 out of 13 companies, they do use visible particles as the other acceptance criterion, where they use absence of visible particles by manual visual inspection. While all of them queried said they do use the subvisible particles following the USP 788 criteria, for which is the light obscuration assay. In addition to that, 10 of 13 companies, they do use MFI, microflow imaging, for product characterization. The next question that we asked the member companies was on the sampling plan that companies apply for testing their admixtures. 8 of 13 companies that responded, they said they use the pre and post inline filters for testing, for sampling, rather, while 7 of them use time course to sample the product for testing, while only 5 of them said they only sample post catheter for product quality. The next question that we asked the member companies is on the testing schedule for the assays, with the exception of the potency, osmolality, and isoelectric focusing, the companies that responded, they all do their assays of the samples within 24 hours of sampling, while the others, they do store the samples frozen prior to testing. Next slide, please. Now I request George Grotz from GSK to discuss the conclusions from this survey. George?
Thank you. Next slide. So in conclusions, the first we list is that the survey revealed diversity in the practices utilized by companies, with variation observed in all key sections, as you heard. Secondly, the diversity may be related to a broad array of molecular modalities, therapeutic areas, routes and modes of administration, stability profiles, and clinical needs. Still, a range of workable approaches that can assist biopharm companies to design and execute studies to meet the needs of their asset and clinical program was identified. And lastly, the results of our study help guide biopharm companies to develop suitable compatibility and in-use studies. Thank you.
Thanks, George. That's lovely. Can we go on to? So I'd like to thank the authors, and you can see them all here, that drafted the publication, and particularly those who've taken their time to present this webinar today.